All right, hello, everyone. Good evening, and thank you for coming to listen to our speaker today. My name is Brett McAllister. I'm the Director of Graduate Admissions here at Nova Southeastern University for Halmos College. Along with my two coworkers, both assistant directors, are Ms. Megan Troy and Mr. Teddy Uy. He's here with me this evening as well. We're here to listen to Dr. Rosanna Milligan. She's an assistant professor with us here at Nova Southeastern University, where she runs our Seascape Ecology Lab. Her research interests lie in understanding how the distributions and biodiversity patterns of deep sea animals are shaped by both natural and anthropogenic drivers across multiple scales. She is particularly interested in understanding how mobile deep sea fauna, particularly fishes, create connections between deep and shallow water ecosystems, how deep sea fishes contribute to the ecosystem's goods and services that we rely on, and how those services might be perturbed by human impacts occurring at ocean depth. Rosanna earned her PhD studying the ecology of deep sea demersal fishes at the University of Glasgow in the UK, before moving to the US as a post-doctoral researcher to study deep sea pelagic ecosystems in the Gulf of Mexico. We're honored to have her. It's very exciting for us to get to listen to all that she gets to work on. And so with that, Zan, we're going to turn it over to you and go ahead and get started.
Perfect. All right, thank you very much. Let me share my screen and we will get this going. Okay, is that looking good? I'm going to assume so, unless anyone tells me otherwise. But cool. Yeah, so tonight I just want to talk a little bit about some of the research that we do in the Seascape Ecology Lab. And kind of focus on a little bit of a case study looking at some of the work we've been doing in recent years in the deep pelagic realm, so the deep water column of the Gulf of Mexico. All right. So what is the deep sea? Well, it's actually the largest habitat available for organisms to live in on Earth. It covers 75% of the surface area of the planet, and it provides well over 95% of the inhabitable living volume available for those animals. So it's a huge realm that, for different organisms to live in. Now, in general, the deep sea is characteristically very energy poor. It's very high pressure, and it's subject to relatively stable physical conditions, which are ideas you maybe are already familiar with. But I don't want to give you the impression that this is an environment is static or in any way unchanging. And certainly the closer we are to the continental shelf break, the closer we are to the surface ocean, really the more dynamic these systems can be. There's a lot of physical and chemical exchange between the oceans and the atmosphere. There's a lot of mixing through deep waters. We have a lot of seafloor water column interactions, and we also have fauna that are moving through this space as well. So on different time scales and different spatial scales, we really are dealing with quite a dynamic environment, subject to lots of different processes. The deep oceans then are highly connected to our other marine ecosystems that we find, for example, in the surface oceans. They're highly connected to coastal systems and even to terrestrial ecosystems through the movement of animals, through the movement of ocean currents, and through the kind of exchange of these physical processes. So kind of when we think about the deep sea, I think very often we think about it as this kind of isolated, out of sight, out of mind kind of system. But actually it's integral, and it's really at the center of our marine ecosystems as a whole, especially this deep water column there. It is kind of the link and the nexus through which all these different marine ecosystems connect together. So understanding what's going on in there is really important to us in understanding marine ecosystems in general. Now, when it comes to understanding marine ecosystems, everything starts at the surface. So the surface ocean is where we have 99% of our marine productivity, far and away the most important source of energy and source of food for the animals that live in marine systems is generated and created in the surface ocean through these little microbes like phytoplankton. They're then grazed on by slightly larger animals, things like our zooplankton grazers, for example, and they in turn are going to be fed on by our larger predators. If you've ever seen documentaries of the open ocean, I'm sure you have seen images like these before, where we have schools of planktivorous fishes like herring and mackerel, for example, being fed on and creating these big bait balls being fed on by diving seabirds, marine mammals, and sharks and things like that. So this is kind of our very surface layer of the ocean now, and this is characteristic of these oceans only in the daytime. And what I want to talk a little bit about here is what's happening below this and how we actually see changes through between day and night and kind of through time as well. So moving deeper then, going down from our surface ocean into the deeper depths of the seas, what we find below all this is a whole range of fishes and crustaceans and cephalopods and other organisms that inhabit the deep waters of the open oceans in the daytime, but then at nighttime are going to come up to the surface oceans, and they're also going to feed on the zooplankton. They're there in those highly productive surface waters. But the reason they only come up at night is so they can avoid those big predators that are feeding there in the daytime. So what we see is this process called diel vertical migration. And this is the largest animal migration on the planet by far. And this is, again, these animals coming up from deep waters to feed at the surface and then return down to depth at night to avoid those predators and stay safe. Now, one of the main kind of processes here that's really important is that these animals, as they come up, they feed at the surface. They feed on carbon that has been produced at the surface, but then they move back down to depth to digest that food, where they defecate, they grow, and some of these guys are going to be fed on by deep living predators. So this whole process, on average, is going to move energy or carbon from the surface ocean down into the ocean interior. And this process through the biological carbon pump, this is the active carbon pump, is a really important mechanism for helping things like in carbon sequestration, for moving carbon out of the atmosphere and kind of locking it into the deeper waters of the ocean. And so this process can kind of carries on down through the water column, all the way down to ultimately the seafloor. If we can get this carbon below about 500 to 1,000 meters or so, we're going to lock that into kind of the ocean interior, where it's going to remain for hopefully centuries at least. So the take home here, and I'll come back to this diagram later, is to kind of really emphasize that when we talk about the deep sea, it is fully connected to the surface ocean, it's fully connected to the seafloor, and it's also connected laterally out to the coasts and shallow waters, and it has these very meaningful effects on how these different ecosystems function and the kind of goods and services that we can derive from them and the benefits. They have a lot of benefits for us as humans as well. But we do need to figure out what those are. So that's what we're trying to do within the Seascape Ecology Lab here, is just kind of understanding the roles, especially of fishes, that they play in these deep sea ecosystems, and trying to understand how they connect with these other parts of the marine realm, and to try and give us ways of better predicting them, so that we can in turn try and understand and better provide management solutions to these kind of pretty poorly known systems. So yeah, what we're really interested in, we're asking a lot of questions related to the ecology and biology of different fish species. We're looking at the distribution patterns of individual taxa and how they relate to biodiversity. And importantly, we're trying to understand and predict how human impacts might be affecting the deep sea as well, in order so that we can better develop these tools for resource management. I'm an ecologist at heart, but a lot of the work that we do has got a very, very strong quantitative focus. There's a lot of data analysis that happens in my lab, as my graduate students will tell you all about. Now, the reason this is really important is, and again, we kind of think maybe of the deep sea as being maybe too far away for humans to have an impact on, but that's not true. And really, as we're kind of depleting easily accessible terrestrial and coastal resources, we're seeing a real shift towards the development of new industries in the offshore realm and in deep waters. And we know now that the deep sea is being impacted by a huge range of human activities, some of which are kind of shown in this figure here. But the main kind of impacts here are through things like climate change. We're going to see shifts in the stratification of the oceans. We're going to see shifts in hypoxia patterns, potential changes to deep circulation. Waste disposal is a huge impact in deep sea systems, both kind of litter and things like that, but also through the potential for carbon sequestration, deliberately being done and using these systems as kind of a dumping ground for some of this stuff. We're also seeing increased exploitation of deep sea resources as well. Like I say, as we're kind of using up easy to access resources, where there's more and more drive to kind of moving further offshore. So things like deep sea fisheries are expanding offshore. There's mining industries now to mine for sulfides and rare earth metals, things like that. And oil and gas, the oil and gas industry is also very much moving further offshore as well to access resources in deeper water, partly because we can and it's economically viable, but also because we have this demand for it that needs to be met. So kind of finding these balances and finding ways of doing this as sustainably as possible is a big driver for the work that I do. But of course, there is nothing is easy. There's always issues when it comes to deep sea. And I kind of love this figure here. So this is taken from a paper published in 2010. But what this figure is showing is the number of species records from different parts of the ocean, where the number of records, the highest number of records is shown in red, and the lowest is shown in blue. And what you can see quite clearly here is that the places that are most accessible in the marine realm have been the best studied. So we know most about the coastal ecosystems. We know most about the continental shelves close to us, and we know most about the surface ocean. Right? These are the areas where we where we know the most about what lives there. But out in the deep ocean over here, especially in the deep water column, we know far, far less. And this really limits our ability to be able to manage these kinds of ecosystems. So filling in this knowledge gap, and this is absolutely the largest habitat on Earth. This is the bulk of the ocean, essentially, is comprised by this deep water habitat. So filling in these knowledge gaps, like I say, is kind of something that we're really trying to do, and that's increasingly important because we have these human footprint. The human footprint is moving further offshore as well. So this all kind of sets the scene. It brings us to what I really want to talk about tonight in a bit more detail. I kind of run through a bit of a case study looking at mesopelagic fishes within the Gulf of Mexico. So again, if you've seen documentaries, Blue Planet, things like that, this is basically all our sea monsters that we find down there. So we have our hatchet fishes, lanternfishes, the classic angler fishes, dragonfishes. All of these guys are the kinds of fishes that I'm talking about from this part of the ocean. So the mesopelagic realm, if you're not familiar with it, this is a twilight zone of the ocean. This is typically defined as depths between about 200 meters and 1,000 meters. It's essentially below the epipelagic zone, which is where primary production and photosynthesis can occur. So we're moving into the zone of the ocean where solar light still penetrates. So we see lots of adaptations for camouflage here, but nobody's producing their own energy. There's no photosynthesis anymore. And so that's why we get a lot of these species migrating up the surface to feed in those energy-rich waters up there. Now, some of the mesopelagic goods and services that we are most interested in include carbon sequestration. This is really the main goods and service that I'm going to consider for the purposes of this talk, and it's probably one of the most valuable goods and services that is provided by the global mesopelagic ocean. So a recent report published 2019 by some people at Woods Hole, some researchers at Woods Hole, estimated that biological processes through the mesopelagic, so this diel vertical migration, is responsible for sequestering approximately 2 to 6 billion tons of carbon per year, with values worth potentially up to almost $1 trillion every year. Now you'll notice in here the estimates of economic value and the carbon itself are pretty variable. These are pretty wide bounds, and these estimates are done at a global scale. So what we really need more of is studies that kind of look at honing in on this variability and better understanding, okay, are there places in the world where this is particularly important? Are there particular species that are very important in providing these goods and that kind of thing? There's also the potential for development of things like economically important fisheries. These are not really taken off in a commercial way as yet, but they are they're kind of always on the radar a little bit there, and there's been a lot more interest in the last kind of 5-10 years or so about kind of developing some of these again, looking at them in more detail. And of course, we have other non-material benefits as well. Things like knowledge creation research is a big service provided by these things as well, which is good for me. But like I say, we're really going to focus on this idea of carbon sequestration today. The case study I'm going to be talking to you about is from the Gulf of Mexico. So the Gulf of Mexico is a semi-enclosed oceanic basin that sits between the Caribbean to the south and the Atlantic Ocean out to the east. And it forms this nice little basin here. It's very much it's kind of US ocean, if you like, to the south of the United States. When we look at the Gulf of Mexico from the surface, what we have here is shallow waters marked in red, and the depth gradient increases to blue as we move into deeper waters. And what it looks like when you look from the surface is it looks like we have a lot of continental shelf. It looks like we have a lot of shallow waters. And this is certainly true if you look at it from above. But if we start to look at this in more in 3 dimensions, and this animation is of course courtesy of Dr. Matt Johnson, who's also Halmos College of Arts and Sciences. You can really see that when we consider the volume of the Gulf of Mexico, over 90% of it lies below 200 meters. Only 9.6% is in this kind of surface and coastal water band at the top. All the rest of the Gulf of Mexico is what we would consider deep sea and greater than 200 meters water depth. So there's a lot to learn down here. The other reason we're interested in the Gulf of Mexico specifically is because back in 2010, the Deepwater Horizon oil spill happened. And this, by many metrics, is now considered to be the worst oil spill in history, or at least the worst marine oil spill in history, I should say, in terms of the amount of oil that was spilled and the duration of it and things like that. And what this really did was, so the Deepwater Horizon oil spill itself occurred at a depth of 1,500 meters and impacted the entire water column. So it's very much a deep sea event within the Gulf of Mexico. The problem is that even though it happened in the Gulf of Mexico, which is really close to the US, very accessible, to a nation that's very good at research and things like that. Nonetheless, we still have very little baseline knowledge of the deep pelagic ecosystem before this event occurred. And so what we've been trying to do in the time since is really understand what happened and kind of piece together that picture. So this is what that oil spill looked like. The wellhead itself was located about 1,500 meters. The blowout preventer failed at the seafloor and then caused the release of all the oil that was spilled over a period of 3 to 4 months in early 2010. Of the oil that was spilled, about 40% of it never actually reached the surface. The rest is believed to have formed buoyant plumes and remained at depth, or kind of sunk out to the seafloor around the area. So, I mean, very significant and hard to detect deep water impacts have occurred from this spill. So without any prior knowledge of what the system was like, what the ecosystem was like in detail before the Deepwater Horizon spill happened, the question really became, well, okay, this disaster has occurred. How do we put that into context? how do we now build up our understanding of the deep water ecosystems of the Gulf of Mexico to kind of better understand what happened, and then to develop predictions of what might happen in the future. So if this happened again, what do we now know about the variation within the system that will help us manage impacts of another oil spill or some other impact in the future? How can we build up that understanding really to help us predict and mitigate and manage the system in terms of what we now know about its vulnerability and things like that? And so the research that I'm going to be talking about is based on 3 major programs that have been underway since 2010. The first of these is the Offshore Nectin Sampling and Analysis Program, which was funded by NOAA's Office of Response and Restoration. This was basically a damage remediation program to go out, and we were trying to understand what could have been impacted. So we surveyed between 2010 and 11, and then conducted the analyses from 2012 to 2015. I should say as well, all of these programs are run by Dr. Tracy Sutton, who's the lead PI on all of these. And also, he runs the Oceanic Ecology Lab within HCAS as well. So our first program was examining what was there that could have been impacted. Our second program is the DEEP-EN program, funded by the Gulf of Mexico Research Initiative. And here we're trying to put our animals into an environmental context. So understanding how variable the system is naturally, what is the role of different ocean currents and water masses, that kind of thing. So how do they influence what's going on? And then knowing that, can we say something about what, whether if we saw anything after the Deepwater Horizon oil spill that was maybe different to what we would expect to see? The program that we're currently working on now is the DEEP-EN Restore program, and this really has a strong management focus. So how do we take all these lessons that we're learning, and how can we really apply these to developing management strategies and working with end users in the Gulf of Mexico to apply these lessons? So again, just to give you some idea of methods, all the data that I'm going to be talking about were collected using something called a MOCNES, or a multiple opening closing net and environmental sensing system. This is basically a type of trawl gear that allows us to catch samples at specific depths. And this is important for us because we have that diel vertical migration. So we need to be able to collect samples at known depths during the day, and we also need to look at samples from known depths during the night, so we can account for that migratory behavior. So we go out and we've collected at this point probably close to 2,000 samples, if not more, over the course of our time series, and we're using those data to really understand and answer our questions. And this is just a few photos on the next few slides of what that looks like. So we have the samples being collected here on the left. This is an example of a lanternfish. Spoilers, this is what we're going to be talking about for the rest of the talk. These types of little fish here. Some are important migrants. But we also catch things like these, and I just love showing this photograph because I think when we tend to think of anglerfishes in the deep sea, we tend to think of these enormous sea monster things, like something out of Finding Nemo. And the reality is that a lot of these fishes are tiny. They're really small. They're like the size of your thumbnail. So I kind of love that. If you've never seen a tiny anglerfish, this is what a lot of them really do look like down there. And of course, we have other fauna as well. We have a lot of shrimps and jellyfish, a lot of gelatinous zooplankton, cephalopods, all these kinds of organisms as well. I'm only going to focus on the fishes because that's what I study for the most part. But again, there are lots of other things down there that are being studied and investigated by other members of the project teams. So, what have we found so far? Well, to date, across all of our samples since 2011, we have records showing that there are about 1,600 known fish species occurring in the Gulf of Mexico. Through our time series of data, we have recorded about 920 of those species from the offshore region. So it's very, very diverse out there, and we find a lot of these different taxa. We've added 180 new species records for the Gulf of Mexico, and we've also added 22 new species that are new records to science. This is all thanks to our taxonomic experts within the program. And we're also finding very strong connections to coastal and benthic systems. So if you kind of recognize these different animals from maybe snorkeling or scuba diving on reefs, or from fishing offshore, it's because a lot of the taxa that we find there, we are also finding in our samples in the offshore deep-pelagic as well. So it seems like this is a pretty good habitat for some of these organisms, kind of places where they can grow up, and we find the juveniles out there apparently doing quite well. Okay, so the bulk of this next part that I want to talk about is, what is the role of these natural drivers? What natural drivers in the Gulf of Mexico are important? And for all of this, I'm going to talk about our lanternfishes as a bit of a case study. The reason for this is because lanternfishes are a highly abundant fish taxa within the deep mesopelagic ocean. They are a dominant part of the micronectin, which means the micronectin are organisms that are between about 2 and 20 centimeters in length. So this is the kind of size range of animals we're talking about and we're sampling for. They are usually number 1 by biomass and number 2 in terms of numbers. We find them throughout the world ocean, so they are globally distributed. Within our data set, we have a really nice sample size to work with. We have almost 32,000 individuals from our data set. The lanternfishes are also really diverse, so we get to look at the biodiversity patterns here and see what's going on with those. Globally, there's about 250 species from 31 genera within the lanternfishes, or Migrators is the family name. And in our data set, we've collected 57 species from 19 genera. So they're, again, very, very diverse in the Gulf of Mexico, giving us a really nice sample set to work with. Overall, the community structure, it seems to be relatively stable over time. So as we kind of look through our time series, we're seeing the same dominant taxa kind of recurring over the time series. And these are the same dominant species that we see referred to in the literature as well from some of the earlier studies from the late 1980s and early 1990s. So this just kind of helps us kind of understand that the communities of fishes that we're looking at, in terms of at least the lanternfishes, they're not changing wildly in terms of who's there and the type of composition that we're seeing. There are other changes that are happening, but we'll talk about those as we go. Lanternfishes, again, are really important. So these are one of our migratory taxa. And they're also, this makes them prey for a wide range of predators. They're relatively robust, fair, kind of high energy prey species. And they're preyed on by things like nocturnally scavenging seabirds, so like Proceliforms, things like shearwaters, for example, petrels. They're going to feed on myctophids at night. They're also preyed on, again, by things like our deep living predators at depth. And then we have a kind of range of oceanic predators that will either feed on them when they come up to the surface at night, or they'll dive down and feed on them where the myctophids are kind of hanging out in their daytime depths. So kind of a variety of strategies, but they are important in the diets of a wide range of different organisms. So like I say, lanternfishes are our migratory taxa. All the species that we know of so far are migratory to a greater or lesser extent. And this is kind of just an echogram showing that same pattern that I showed you before, where the lanternfishes are going to stay at depth during the daytime, and they're going to come up to the surface ocean to feed at nighttime. Because they are voracious predators of zooplankton, they are also really important in this active carbon transport. So they're going to consume a lot of energy from the surface, and they're going to move that down into the ocean interior. Now, the big questions for us are, while we know something about who lives in the Gulf of Mexico and the kind of behaviors they exhibit, we don't know kind of where they're distributed or what the variation in those distributions looks like. Are there times of year where we see more or less of them, that kind of thing? Is there, what are the influences of the environment on them? So this kind of variation is something that we're really interested in disentangling. So like I say, our lanternfishes are migratory, and this figure on the right is something that I just kind of want to run through with you, because I'm going to show you a lot of figures that are going to look like this. So what we have here on the right-hand side is a bar chart or box plot that is essentially running from 0 meters, so the surface is at the top of the y-axis, running all the way down to 1500 meters, which is the maximum depth of our samples. And it's divided into a range of depth bins corresponding to those, the depth bins that we sampled with our nets. The white bars are show the distributions of the lanternfishes with depth during the daytime, and the black bars show where they are during the nighttime. So in this example, you can see here that we have about 85% of the lanternfishes occurring between 200 meters and 1,000 meters during the day in the white. But at night, we see about 68% of those in total, moving up to the epipelagic realm. And that's basically corresponding to this migratory pattern on average, right? So I'm going to show you a number of figures that look like this. You're going to see this type of pattern, this type of chart repeated in the next few slides. Okay, so we have our lanternfishes. We know they're important in moving carbon through the ocean. How do we start to think about and predict their vertical distributions? Is there anything we can see here? So the pattern I just showed you is kind of the average pattern for the family within all of our samples. But when we break down these patterns and start looking at them species by species, which you can see in the charts here, we have the top 15 or so species, they all show quite different rates of migration. They have different daytime depths that they're preferentially inhabiting. And so there's quite a lot of variability in here between the species. And you can kind of see that if we look at just this middle row here, you can see some of that variability here where we have Hygophilum benoiti on the left-hand side with a mean migration rate of only 20%. Species like Notoscopulus resplendens in the middle has a mean migration rate of about 90%. So all of these members of the population essentially are migrating up every night. And then we have species that are a little bit more in the middle, like Myctophilum aphene, for example, which has a migration rate closer to 50%. So sometimes they migrate, sometimes they don't. And this kind of variability is what we're really interested in. And kind of asking these questions are there rules that the different fishes are following that we can predict to kind of better understand what's going on? So to kind of help us here, we can think about migrators in different ways according to different categories. So the first category here is we're going to consider complete migrators. From our point of view, these are not particularly interesting, but these are species where the entire population migrates each night. It implies there's something kind of intrinsic or hardwired in these species that requires them to migrate every night. Whether this is something like an endogenous circadian rhythm, for example, or whether it's just that, you know, they have to feed every single night no matter what, so they're going to migrate every day. You know, something like that is driving them. There's no variation here. If there's no variation here, then that's, you know, there's nothing really there to explore. So we're not going to really think about these guys too much. Similarly, we have other taxa that are non-migrators. We don't see this in the lanternfishes, but it's the same idea. Basically, the distributions in the day are the same as at night. Again, not a lot of variation here for us to get our teeth into and try and explain. But the group that we really are interested in are a group called partial migrators. And this is where only some of the population migrates each night. It implies that there is some kind of extrinsic cue that is cueing the behavior, whether it's environmental or you know, related to satiation or maybe a breeding cycle, something like that. But there's variation here that we are interested in explaining, and that's what we're going to think about. So the key here is that these partial migrants, again, are not a uniform group, and it seems like there's lots of variability between these different species to look at. So when we're thinking about what drives the variability, there's lots of different options here. We're going to think about environmental variables like water mass structure. That's going to be the main focus that I'm going to talk about tonight. But there are others as well that we're starting to think about. We know that lunar phase is important for some species, for example, and we've just had 2 master's projects finish up work looking at how the vertical distribution of different species varies with size and age of the animals, for example. So that's going to be coming out soon, I hope. But again, for today, I'm going to focus on this environmental questions or case study. So in the Gulf of Mexico, we have a couple of different water mass types that we have identified. Now you can see the animation on the left-hand side here is showing water basically coming in from the Caribbean into the Gulf of Mexico. Now we call this the loop current. And so we identify loop current origin water, or LCOW, as our water mass that represents the Caribbean sourced water. Now this is different to Gulf common water, which is kind of our background water mass within the Gulf. The 2 water masses are quite distinct from each other. Loop current origin waters have generally low productivity. They're very clear. And so we get a lot of light penetration through this particular water mass. It's a little bit warmer than surrounding waters as well. And generally has been associated with reduced biomass of fishes and zooplankton compared to Gulf common water, which we find in the rest of the Gulf. And so when we start to look at kind of, again, overall patterns here, just looking at all the lanternfishes together, we start to see a couple of rules starting to emerge. So here we have 2 of these T-plots. We have Gulf common water in gray, the gray panel on the left-hand side, and we have loop current origin water on the right-hand side. Now, in both cases, you'll notice in the daytime, about 85% of our myctophids are found, or lanternfishes are found in the mesopelagic, but in the Gulf common water, most of them are found in the upper mesopelagic between 200 and 600 meters, whereas in loop current origin water, we find most of them in the lower mesopelagic, so 600 to 1,000 meters. And this is seems to be a fairly consistent pattern that we see. We also see differences in migration rate. In Gulf common water, the migration rate is approximately 71%, whereas in loop current water, it overall reduces to about 44%, suggesting that, again, there's maybe something going on here. The issue we have, of course, is that we can look at this, these rules kind of in overall terms, but again, when we break it down and look at individual species, we find that different species are not necessarily following all of these rules, but or they maybe only choose to follow some of them. But nobody is doing anything different. They're either not obeying these rules, or they are doing some variation on these. So I'll show you what I mean by that. So for example, this is Stratoscopulus warming eye. This is our most abundant lanternfish in the Gulf of Mexico. And it shows really no difference in patterns between common water and loop current origin water. Now, what I've done with these charts here is I have separated out the daytime depths only on the left-hand side, and I'm showing the nighttime plots on the right-hand side. So you can more clearly see the difference between loop current water in gray, Gulf common water in gray and loop current water in red here. So in the daytime, there's not really any differences in the proportional abundances. Same at night. These guys, they don't show any difference in the patterns. For our second most abundant species, they follow one of the rules. So they show vertical displacement during the day only, but there's no change in migration rates. And then still in other species, we see kind of extreme versions of both, where we see vertical displacement during the daytime, and we also see a complete inhibition of migration during the nighttime as well. So these rules seem to be consistent from what we've seen across kind of all the fishes. These are the rules that are being followed by, in terms of vertical distributions within these 2 water masses. But different species are going to kind of pick and choose which ones they're going to follow, if any. Now, the reason we think this is happening is, I think, related to something called light comfort zones. Mesopelagic fishes are all evolved to hide from predators, and in that mesopelagic zone, there's still solar light. that can illuminate you. So they've evolved a number of different strategies, like counter-illumination, with different forms of camouflage, for example, in order to really blend in with their surroundings. Remember, in the open ocean, there's nothing to hide behind. You are completely out in open water. So you're either going to camouflage yourself or you're not. There's no coral reef to hide in, for example, or boulders to hide under. And so there's this idea then that different species have evolved to be better adapted to different light levels. And this is kind of called their light comfort zone, if you like. And so because we have this different, the different properties of the water mean that loop current waters are clearer and less turbid, light can penetrate much deeper, which would explain why in the daytime we see some of these species moving into these deeper depth bands. Of course, if you move deeper, that also means that you have to travel further if you're going to migrate to your feeding grounds in the epipelagic. So it's potential then, there's a potential then that we have this trade-off between where you can best hide from predators, and then is it worth the metabolic costs of migrating to the surface to feed, or are you better off skipping a meal, say, and not migrating under those conditions? Again, we have this kind of these overall rules, but I think the idea then that we have these different species adapting to their own niches kind of gives us this idea of why we would then see all these different patterns in vertical distribution. The next very short section that I want to talk about is how we predict changes in the horizontal distribution. So this last section obviously is thinking very much about how they're structured vertically in the water column. And this next section is going to be much shorter because the kind of short version of this is that our assemblages of lanternfishes seems to be very well mixed at horizontal scales. We see, at least the community level, we see some evidence of things like loop current water maybe having a weak effect, suggesting it maybe influences some species but not others, which again kind of is what we've seen with the vertical distribution patterns. But over the horizontal scales that we've been studying, there's really very little difference in species composition across the board. And what we think is essentially happening here is that what we're looking at is a very well mixed kind of blended assemblage where we have all these different species following their individual behaviors. They're staying in their preferred depth range at the daytime, they're migrating in their own individual ways. When you take that and you combine it with the fact that we have different currents and different current speeds moving at different depths, you're going to be moving the different parts of this assemblage around the Gulf in different ways. As they migrate, they're going to be exposed to different currents. They're going to be swirled up and move passively, potentially over fairly large horizontal distances. So it seems that the story here with these guys is that the vertical distribution and vertical migration is really important, but passive dispersal is the thing that's driving them across horizontal scales. So what we're seeing here then, to kind of summarize this little case study, is that we see a lot of different variation here in the fish behaviors influenced by the very dynamic environment that they inhabit. And what this is starting to give us is a little bit of insight into how this could be, how their behaviors could influence things like carbon sequestration and the biological carbon pump. And it really gives us kind of vital context for these human impacts. When we are thinking about how we manage ecosystems and things, I think one of the big questions for us to answer next is going to be things like, well, do the general patterns explain enough, or do we really have to dive into these individual species patterns to understand the system? Where is the line where enough is enough kind of thing? But speaking of that vital context, what about Deepwater Horizon? So this was kind of the impetus for all of this work at the beginning. And one of the things that we've kind of been trying to do is to put that spill into context by looking at all these different avenues of work and these different research questions, and really kind of say, okay, like I said before, did we see anything from Deepwater Horizon oil spill, and did it fall within the natural bounds of variation in the system, or is it different? So by taking all of this into account, what we can do is kind of run some sensible models and models that allow for this very natural variability as best we can. and then kind of look and see what changes have occurred. And one of the things we have found is that the abundances of all our deep water fauna, our deep living fauna, has declined by somewhere between 50 and 75% between the 2011 data set and between our later data sets collected through DeepEnd. Our lanternfishes have been quite badly impacted here, and they've declined by about 85% across the board. So there's been a pretty big decrease in just the numbers of organisms out there. And of course, the question then is, well, was this caused by Deepwater Horizon? And the one thing I will say to that is that we don't have any pre-spill data. So just based on these findings, you know, we can never say 100%, yes, this was caused by the spill or not. What I will say, though, is that we are still detecting pollutants in the tissues of some of our organisms. This is work being done by our colleague Isabel Romero on the west coast of Florida. And there's no other kind of big smoking guns, environmentally speaking, that kind of point to anything that kind of occurred or would explain this magnitude of a decrease at the same time. It's not to say there isn't anything, but you know, that's kind of where we're at with that just now. And again, we're working this out at the moment for publication, so we're kind of in the last stages of analysis here on this. The question here, I think, before I move on, is when we think about impacts on ecosystem functioning and the carbon transport and things, what we have here is a huge decline that's occurred across our deep living organisms. And I guess a more interesting question than was this caused by Deepwater Horizon is, well, maybe if something like this happened again, if an impact of this magnitude occurred again, regardless of what caused it, what are the impacts then on things like carbon sequestration and the other ecosystem goods and services provided by the system? And again, this is a whole avenue of research, I think, that we're really interested in exploring. And I've got a couple of students working on topics in that vein just now. So just to round us up then, just to kind of give you an idea, again, of some of the kind of future work and future avenues and the kinds of student projects that we have going on in the Seascape Ecology Lab. We're going to be focusing in on some of our indicator taxa. We have a handful of species that we really want to focus on and get a better idea of what they're doing, see if we can use those as proxies for the rest of the ecosystem and other taxa. We're exploring species distributions and new predictors. So we're looking again in more detail to kind of better understand, you know, do we need our general patterns okay, or do we need really the species level distinctions, or even less than that? Do we need to know whether we're looking at juveniles or adults, that kind of thing? And we're looking at different predictors as well. So we have a project by Maida Hughes at the moment, for example, looking at whether we can use satellite estimates of light intensity to model where our fishes are. We're going to be extending our analyses to account for animal sizes and life histories. We have some projects underway at the moment that are going to be extending our work, but using abundances out to biomass and body size analyses. And we're also going to be looking to develop specific models to really focus in on this carbon flux question as well. So we have a couple of projects just rolling up at the moment, getting those started and underway just now. We're also actively collaborating with resource managers within the offshore Gulf of Mexico. So working with the marine mammal group, for example. Marine mammals are a major predator of some of our larger species. And so we're kind of working with them to see if we can figure out, well, if we can predict where our prey taxa are, can you guys use that to predict where your marine mammal predators are going to be, and that kind of thing as well. So we're working on a whole bunch of different avenues like that as well, really trying to get this work out there and get it used. So just to summarize everything here, I know we're kind of getting close to time, but just to kind of summarize up, what I kind of want you to take from this is this idea then. Our deep sea ecosystems are highly connected to each other and to maybe the coastal and surface ecosystems that we are much more familiar with generally as humans. By understanding the context in which the natural drivers occur and how these connections occur and change through time, we're going to be in a much better place to be able to explain and provide context to human impacts as they occur and potentially do something about those as well. Of course, this is all just scratching the surface. We still have a lot of questions that we want to ask, and I just kind of want to leave you with this video here just to highlight that. This video has absolutely nothing to do with me, but it is a video that was filmed in the Gulf of Mexico a couple of years ago during a research cruise. And it's the first footage from or anywhere outside of the Western Pacific, I think, of a living giant squid underwater. And this, again, was filmed in the Gulf of Mexico. We had no idea that that was there until very, very recently. So plenty out there still to be discovered and plenty of research questions to be asked and answered. Cool. So with that, I'm just going to say thank you for taking the time to listen to me today. Thank you to everyone, of course, involved in the research, and all the photographs that you saw today of our deep water fishes and fauna were taken by Dante Fenolian, who is our resident photographer. Okay, thank you very much. I'll be happy to take any questions for as long as we have time.
Absolutely. Zan, thank you so much once again. As far as questions go, if you guys wouldn't mind just typing them in the chat, I'll kind of moderate back and forth and help Dr. Zan. So feel free to ask any questions that you may have. Well, I know, Zan, that I do have a couple. Maybe you scared him a little bit too much. You're talking some of the stuff. So my big question is, I know obviously I see you guys every day at work, but from how often do you guys get to go on these research cruises, as you guys have spoken about on numerous times? How often do you get to go out and do this like hands-on research?
So it depends a little bit on the program. Through Deep End, where we're going out twice a year. Through, all right, that's the dogs are getting rowdy. Now we've just been out in August there. We're going to be going out again in May next year. It's a little bit program dependent. Obviously, getting the boats out to go offshore and work in deep waters is pretty expensive. So yeah, it's a little bit more limited than popping out to a local coral reef or the mangroves or something.
All right, well, Professor, we don't want to hold you up anymore. I'm sure you got plenty of more work to do with everything. So for those of you who came this evening, thank you so much. We'll be reposting this again on YouTube at a later time once we get it all downloaded. Please feel free to share it with any friends, coworkers, anybody. I'm just happy to get this amazing information out there. I know I learned a lot, as I always do whenever I listen to you guys talk. So Zan, thank you very much once again. It's always a pleasure. Hope you have a wonderful evening. Take care.
My pleasure. Thanks for having me. See you.